Welcome to the Reproductive Omics Tech Laboratory, celebrating 10 years of groundbreaking research and discovery. We're excited to share an in-depth look at the focus of our work, our innovative lab processes, and the dedicated team driving our success. Over the past decade, we as the laboratory made significant strides in understanding cellular behaviors and advancing the field of omics. Everything began with these two science-driven and successful professors. Everything begins here, in the Laboratory of Reproductive Omics Technologies. Our research begins either with cell lines, the foundation for studying complex biological processes, or with tissue that has to be first dissociated. We use our tissue disruptor, MediMachine 2, to prepare samples by homogenizing tumor biopsies or clinical tissue samples. This ensures that proteins, nucleic acids, and other biomolecules are efficiently extracted for further analysis. The cells can be then recreated in a complex 3D environment to study the interactions between different cell types within a more complex, multicellular environment, or used for bioengineering purposes, for which in our laboratory we use the extrusion bioprinter by CellInk, BioX, and the inkjet nanoliter dispenser by Biofluidics, BioSpot Arc. The cells can be also integrated into a microfluidic device and observed under controlled, continuous media flow using the microfluidics system provided by L-Flow. This allows us to simulate real-life physiological and pathological conditions. What about if we want to explore these cells with more details? In the lab we have simple solutions for quick observations like the inverted light microscope by Modic, and the fluorescent microscope, Ataluma, for live imaging in the incubator. But we're also equipped with cutting-edge technology such as the spinning disc benchtop confocal microscope BC43 by Andor, thanks to which we can perform time-lapse imaging. This enables us to track dynamic changes in cell shape, migration, spreading, and adhesion. For high-resolution 3D imaging, we rely on the NanoLive 3D Cell Explorer, a label-free advanced microscopy system. This tool is revolutionary for studying live cells in real time, without fluorescent dyes or labels. OMICS technologies are primarily aimed at the detection of genes, epigenetic modifications, mRNA, proteins, and metabolites. For the first three levels of detection, we have implemented the third-generation sequencing platforms of Oxford Nanopores, Grid-ION, and ProMeth-ION. We can perform whole genome sequencing, transcriptome analysis, epigenomics, and single-cell sequencing. On every step, of course, we perform a fragment quality check using the Biofragment Analyzer, QSEP 100, provided by Bioptic. But what if we want to explore the proteins? For detecting and quantifying protein expression and post-translational modifications, we use the simple Western blot platform, JES. It enables us to quantify multiple targets in a single sample. For cell characterization and quantification, we use the Sony cell sorter, which allows us to identify and isolate specific cell populations based on markers on their surface. We are aware of the importance of implementing automation in the lab workflow, which will eliminate human errors and biases. For this reason, many of the protocols in our lab can be automated using the OT2 liquid handling system by Opentron to enhance efficacy, enabling us to scale processes without compromising accuracy. Our lab is driven by a dynamic, talented team of doctorants and postdoc researchers led by Professor Soren Hyrabedian and Professor Krasimira Todorova, who have been recognized with numerous awards and honors in their fields. We are focusing on important health problems and topics like infertility, cancer, cardiovascular diseases, and metabolic diseases, and we are winning acknowledgement not only in the scientific community, but on a society level as well. The motto of the lab is to constantly evolve, be innovative, think out of the box, and contribute to science and the community. Thank you for joining us in celebrating a decade of innovation and discovery.